Hello and welcome to another enriching session of Recitals in-Depth by Vajiram Ravi. In this session, we will discuss monthly compilation of all the important current affairs from the month of June 2024. Broadly, we will be learning about the discovery of new virus and genes, the eco-friendly satellites and missions, innovation in biotechnology, biofuel economy and associated innovations, some harmful chemicals and deadly diseases. And we will also see India's contribution in science and technology. So let's begin the discussion. Hello, and now I welcome you to the second part of Science and Tech discussion from the month of June. These are the topics that we shall be covering. A very quick overview about them. First of all, we are going to read about what are reverse transcriptase enzymes and how are they beneficial in helping not just bacteria to fight against the bacteriophage, but also helping us to fight COVID because of some of the diagnostic tests like RT-PCR made on similar principle. The second one is an ISRO mission, the TRISHNA Mission, which is basically water mapping of the land resources and the water resources. Second is the gene-derived technology, a very interesting one after the Volvatia method in order to control the population of mosquitoes. Because it has been asked in UPSC already, this technology we shall be reading thoroughly. The next one is a very unique type but hypothetical gene. This is a green beard gene and how it shows nature's way of altruism. We'll be reading that. Then we will be looking into a sugar-free or a dyed form of sugar. This is the Xylitol, majorly used in your chewing gums and your dental care products. Next is we will be finding some of the craters on Mars surface that has been found by the Physical Space Laboratory of India. Then do you know that some very resilient bacteria have been found on the International Space Station? Well, they are the Enterobacter bugandensis. The next one is Streptococcal toxic shock syndrome that has shocked the population of Japan because of its varied prevalence. Next one is methanol, and we shall be discovering methanol economy, especially with respect to India. We will also be reading how methanol is causing a lot of deaths in India. Then we will read about Anthropic's large language model, just in line with the ChatGPT and the Bard AI, and this is named as Claude Sonnet. And lastly, we will be reading about superhydrophobic catalyst and how are they very instrumental in putting impetus towards the biodiesel economy or towards the bioethanol economy of India. So let's have a detailed discussion now. Now, picking up from the first topic. The first topic is about reverse transcriptase enzyme. This word must be coming out very familiar to you because in the first discussion, in the first slide, we have already discussed about what is transcription and reverse transcriptions. What are the bacteria or what are the viruses which shows transcription properties and which of them shows the reverse transcription properties. So this topic must be familiar to you. Now let's start with the context. There are two contexts. First of all, some researchers have evidently shown that with the help of these enzymes, which are the RT enzymes, some bacteria were able to find their defense against the bacteriophages. What are bacteriophages? I have told you time and again, bacteriophages are the ones who eat bacteria. One who eat the bacteria. These are the bacteriophages. Naturally, the species is not bacterial in nature. Bacteriophages are virus. What we can see is with the help of RT enzyme, researchers have found that some of the bacteria have made or created an antimicrobial resistance or a kind of defense protection against these bacteriophages, right? One more question. Does the bacteriophage can also eat up the human cells or can they feed on the human body? Can they infect the human cells? That is a question which I want you to answer in the comment section in order to understand how much in depth you have learned about bacteriophages so far. We have already learned that bacteriophages are one of the most prominent reason as to why Ganga has been considered as one of the most purest rivers. Why? Because of concentration of bacteriophage only. But any whichever way, you should also know this is the most abundant form of viruses available across the ecosystems. Coming back to the context, the first context is researchers have explored this, and the second context is that its wide application has also been found. Application of what? The reverse transcriptase enzyme. Its application has been found in order to diagnose the SARS-CoV. Have you heard about the RT-PCR test? Well, in the RT-PCR, RT reflects for reverse transcriptase only. How was it useful? We will be learning eventually. But first of all, let me explore with you what is reverse transcriptase enzyme. First of all, if any biological molecule or its nomenclature comprises of something as is in the suffix, it suggests to me that this is an enzyme. For example, amylase, for example, peptase, all of those are enzymes. These are not acids as such, okay? So this is first of all an enzyme. This enzyme would definitely be facilitating the production of something, or it would facilitate reaction of something. What is this reaction? The reverse transcriptase enzyme is basically a DNA polymerase. It's basically a DNA polymerase enzyme. What do I mean by that? DNA polymer is created with the help of this RT enzyme. Now, to make you understand it better, why do we need to create DNA in the first place? And is it even possible that I am able to create DNA from something? Yes, and it is possible with the help of RNAs. But ma'am, in the first class, in the first topic you told that DNA can be converted into RNAs. And this process was known as transcription process. Why? Because the script is getting changed. Because there is a difference in the structure of DNA and the RNA. As you can see from the classification, I will give you a very brief classification of DNA and RNA. DNAs are more complex polymer compounds. They basically contain the entire genetic code of an organism, especially the eukaryotic complex organisms. Some of the organisms, for example, bacteria, virus, can also contain RNAs. There are different classification of virus, hence, as we have seen in the Baltimore classification. So, DNA is a double helix structure comprising of 4 base pairs. 4 base pairs, and the way they are arranged makes all the genetic information encoded, and it makes all the DNA footprints unique in themselves. What are these base pairs? When it comes to DNA, it is A combined with T and C combined with G. Adenine, thymine, cytosine and guanine. This is DNA structure. How to remember these base pairs? This is apple under a tree and this is car inside a garage. Now, will you able to remember this? When it comes to RNA, first of all, it is not deoxy. It is just ribonucleic acid. It is not deoxy unlike the DNA. The second thing is, in here we will replace this thymine with uracil. In the case of RNA. RNA is more simple. It has got single strand. This one has got double strand. Of course, when it comes to stability of these two molecules, which one will be more stable? This one will be more stable. This one will be rendering lesser amount of mutations. We've already discussed it in the first class. And what about RNA? This will show more number of mutations and this will be less stable. Please understand all these properties because I am building a foundation for you to understand the significance of this enzyme. Okay, now that you have understood that DNA can be converted into RNA, and this happens in all the molecules, in all the cells. This happens in all the cells and this process is known as transcription. But what if some of the molecules render property where they can create RNA, where they can create DNA from RNA molecule?
If I want a simple molecule to be converted into a more complex genetic code, I definitely would be requiring something which could facilitate this reaction, because this is not a natural process. Natural process is the breaking down of DNA into smaller RNA. But if I want to reverse this natural process, some intervention is required. What is that intervention? In chemistry, we call it the catalyst. In biology, we call it the enzymes. So these enzymes, they cause reverse transcription, because now it is getting generated from RNA to DNA. So the entire idea, the entire objective principle behind reverse transcriptase enzyme is basically to perform the reverse transcription process and to create DNA molecules from the RNA molecules. Now you will ask, how is it important and why this RT enzyme is gaining a lot of momentum these days? There are multiple reasons behind it. First of all, you need to understand the difference between the properties of RNA and DNA. I have very briefly told you about it, which one is more stable. If I want to handle something easily, then it will definitely be DNA, because it is more stable, right? So now, let's say, first of all, I want to create a vaccine. Now, a vaccine that is RNA vaccine and a vaccine that is DNA vaccine, which of them would be more stable in nature? Of course, the DNA vaccine, right? I can very easily create the DNA vaccines, but vaccines are created from the causative agents also. What if the causative agent is an RNA virus? Can you name some? We get hepatitis B, the HIV AIDS, the coronavirus were caused from the RNA viruses. If I want to create DNA vaccines, how will I create? I have to do reverse transcription. So first application is creating vaccines. So basically, it will help me in disease management. The first application is disease management. Now, second application is diagnostics. For example, the RT-PCR test. So basically, this polymerase reaction test is based on a principle. In order to identify any enzyme or in order to identify any particular virus, I first of all need to multiply it number of times. Ideally, we have this formula, but only for the DNA samples. We can only cut the DNA samples and create multiple copies of the same, because RNA would be very fragile. It will not be able to give a lot of copies. So again, in order to diagnose the disease which is caused by RNA virus, I again need to convert it into DNA format. So again, diagnostics would include the same polymerase chain reaction where reverse transcription enzyme has been used. So basically, let's say there is coronavirus. So this coronavirus is an RNA virus. Please remember, this is a very important fact. It's an RNA virus, not a DNA virus. What is a DNA virus, by the way? Again, we have read it in first topic. Let's say the adenoviruses, they are DNA viruses, okay? But these RNA virus, if I want to detect it, first of all, I need to create DNA copy of it. So I will be creating DNA copies. These DNA copies will be multiplied n number of times, and then they will be checked with the help of a microscope. And this is how I will be able to perform the RT-PCR test. So it is also very important in diagnostics. Third application. Now, can I also not use it in order to deal with the antimicrobial resistance, which has, by the way, been touted by World Health Organization as one of the top 10 most destructive diseases of the world. So antimicrobial resistance, it basically not only impacts the humans, but also the animals, but also the plants' entire biodiversity is impacted by it. You have already seen that with the help of this reverse transcriptase enzyme, some of the bacteria who were basically the RNA bacteria, they converted some DNA codes, and they started inheriting it into their family line, with the help of which they could fight against the bacteriophage virus. What are they essentially doing? They are creating DNA copies which are antimicrobial resistance. So if I am able to understand this will also reflect how evolution happens, how survival of the fittest happen by generating the right amount of DNA, which can be permanently there in all the generations, right? It is unlike RNA, which is very unstable. So I am creating a very stable copy of something, and then I want the entire family line to inherit it. Basically, it will facilitate the evolutionary studies. So if I know good things, I have good understanding about this enzyme with the help of research and development, I will be able to understand how evolution took place, how survival of the fittest was manifested in the nature, right? Coming back to the point. So I was saying that this is antimicrobial resistance which it has generated. Now, if I am able to understand this enzyme, I will be able to generate certain DNA codes which can fight off this AMR as well, right? So it can also help me to fight against the antimicrobial resistance and the multi-drug resistant diseases. These two are the biggest menace. Antimicrobial resistance is not just against bacteria but also against all the microorganisms, all the pathogens, for example virus, fungi, etc. Antimicrobial resistance means that the pathogens who were causing the infectious diseases, they could not be killed because the drug that was killing them, the antimicrobial drug that was killing them, is rendered ineffective because these bacteria, these pathogens have created an immunity around it. Therefore this is known as the antimicrobial resistance. It renders curation of a lot of diseases, for example hepatitis, HIV/AIDS, for example typhoid, for example TB. All of them are rendered ineffective. The medication rendered ineffective because of development of AMR in the patient. Okay. Now, the other thing is, this is a central dogma which we read already. What is this transcription process? What is translation process? So basically, how does one genetic code translates into a protein and finally manifests its role in the body? This central dogma suggests to me that there can only be a unidirectional flow of information, which means the origin would always be a DNA, then it will flow towards RNA, then it will flow towards protein. But this particular finding, the RT enzyme finding, these researchers also suggest to us that there can be a bidirectional flow of information. So this is also validated with the help of these enzymes. Talking about the research and development of the enzymes, it also eases the R&D. How it eases the R&D? Because let's say the researchers want to test something which is having RNA encoding, but RNAs are very fragile, very difficult to handle, very difficult to have researches on them, very difficult to replicate them. In order to understand the RNAs better, can we create DNA copies? These are more stable copies and they can be read more easily, researched more easily for a longer period of time because of its durability. So it also supports the R&D in the field of biotechnology. So I hope you have understood what is reverse transcriptase, whether it's an enzyme or whether it's an acid. You've understood what is the process of transcription and reverse transcription. You've understood what are the multiple applications. You have understood what are bacteriophages. You've understood the technology behind RT-PCR and where was it used. So basically with this we will wind up this particular topic. The next topic is about TRISHNA Mission. So recently, in the month of June, we also celebrate the World Environment Day, which is celebrated on 5th of June. In that particular day, ISRO announced the details of the new upcoming mission that ISRO is launching along with the French Space Agency. And this particular mission is known as the TRISHNA Mission. The basic objective is to understand the energy budgets and the water budgets all across the earth. How will it take place? Let's first understand the expanded name of TRISHNA Mission. First of all, it is an imaging satellite. Imaging satellite of which form? It is an imaging satellite working in the thermal infrared spectrum. And what is the objective? For assessment of high resolution assessment of the natural resources. and this itself is the full form of TRISHNA Mission. Now, let's quickly have some of the good points about it. First of all, who created it or who is behind the mission? It is a collaboration between two space agencies. One is the CNES, this is the Space Agency of France, along with ISRO. So both are coming together in order to create a satellite which can bring very high resolution for both the space resources and the water resources, for example the coastal areas, the oceanic areas, etc., with the help of which we will be able to get very high resolution. And this very high resolution will be helpful in what? In attaining the objective. What is the objective? The objective is to calculate the overall energy budget, and also, as the name suggests, Trishna means thirst, and thirst is basically longing for water. So the second aspect is to observe the water budget of the earth. How will you observe the water budget of the earth? First of all, it will be able to calculate the amount of clouds, the amount of evapotranspiration that is taking place from the surface of the earth, from plants, etc. Then it will be able to observe how many reservoirs are there, what are the river systems, and not just that, how is water used, what is the amount of water usages, and what are the differential usages of water across the world. So it will also be reading about, it will also be looking into the uses of the water. Then it will also look into the droughts that are prevalent or that can be there in some parts of the earth. It will also be learning about some of the hidden sources of subsurfacial water. So basically, an all-in-all budget of from where the water is coming and how is it going will be calculated with the help of this Trishna mission. Why? Because of a very high resolution. The second one is it also calculates the energy budget. Energy budget, by the way, I have already taught you about the radiation budget. Now, can you tell me in the comment section that in the first part, we read about a particular mission which is responsible for carrying out the radiation budget of the earth, especially the impact of cloud on the radiation budget. So please tell that in the comment section. Now, coming to the energy budget, in this we read about the areas which are having high energies. For example, geothermal provinces. Geothermal provinces have very high energy. Regions where volcanoes erupt, they have very high energy. Surprisingly, some of the urban areas, for example, the urban heat islands, you must have already heard about it, they are also sources of high heat surplus. So they will also be read. So basically, with the help of a multiple spectral resolution, they will be able to calculate the amount of water and the amount of energy budgets in the earth. Now, coming back to the main point, see, how are they going to do it? How are they going to do it? With the help of two payloads. So, this resolution can be achieved with the help of two payloads. One payload, the thermal infrared payload, is created by France. And the second payload, the second which is near infrared and the infrared payload is created by India. When we read the spectrums, you must have already heard about what is infrared, what is visible range. So across the visible range and the infrared range, we are not going to utilize UV rays, X-rays over here. So please remember, for imaging purposes, we generally use the infrared rays and the near infrared rays and sometimes the visible rays. Okay, so for these two things, two payloads have been created by France and India individually. With the help of this, a very high amount of resolution will be obtained, which will be, if you talk about the resolution, it will be about 1 kilometer for the oceans, and it will be about 75 meters for the land. Such a high precision will be stricken. With the help of this, we will be able to understand the water security issues. We will be able to have better data about the droughts that are prevailing, the upcoming droughts, how much water is being used in irrigation, so that we'll be able to understand the food security, the desertification, the amount of agriculture that is creating or that is consuming or having a high water footprint. So basically, with the help of this, a lot of climate challenges, food security challenges, water security challenges can also be addressed with the help of valid data. Now, from prelims point of view, these are the few things which you must understand from TRISHNA Mission. So first of all is, I have already told that how the frequency and the wavelength varies in a spectrum. So in a spectrum, we have basically the frequency starting from the highest frequencies of gamma, and this frequency is always inversely proportional to the wavelength. Okay, so higher the frequency, lower the wavelength, you all know about that. In this particular case, we are going to deal with the infrared and the visible range. I have already told you this. So they basically come in a range where the wavelengths are high. The wavelengths are low, frequency is extremely high in the X-rays and the gamma rays. These are basically emitted from the spatial objects, for example, the black holes having very intense energy, neutron stars, etc. Okay, and the highest of the wavelength is carried by the radio waves, which is used for communication. Okay, now the next thing is the data that we are going to incorporate from the TRISHNA Mission that can be useful in many areas. First of all, in addressing our SDG goals regarding food security, water security, climate resilience, etc. But not only that, 2 more initiatives, global initiatives, will be taking benefit of them. From prelims point of view, they become very important. First is, this data of TRISHNA Mission will be incorporated in the GeoGLAMS initiative. It stands for the Group on Earth Observation Global Agricultural Monitoring Initiative. This was launched in the year 2011 by whom? Surprisingly, by the agriculture ministers of the G20. Right? So G20 has launched the GeoGLAM mission, under which the data on agricultural monitoring will be open sourced and it will be available to all the member countries in order to deal with food security and the agricultural efficiency use of water. Okay, so basically we are mapping the entire water budget, so why not use it for the GeoGLAM data? So we'll be providing that. The next place at which we are going to use the data is the Global Water Watch. Of course, because we are going to analyze the water budget, we are also going to incorporate it in Global Water Watch. Global Water Watch is not an intergovernmental initiative. As you can see from the Water Watch, that it collects the data regarding the reservoir, how much water is there in the reservoirs, in the river streams, etc. in the subsurfacial water. But the most important thing is that it is founded by the World Resource Institute and World Wildlife Fund. Very surprising organizations, but these are the organizations. First of all, this is not an intergovernmental, unlike the GeoGLAM, and this one is created by WRI and WWF. Okay. Nowadays, due to climate change, rising population, urbanization, mismanagement of the waste, takes place rise of another thing, which is the mosquito-borne diseases. And this is especially a menace for the tropical and the subtropical countries, especially countries like India. Therefore, topics related to mosquito-borne diseases, their transmission, their prevention also becomes important from UPSC point of view and for general knowledge of Indians. Here in this theme, we will be reading about the gene drive technology. As the name suggests, something related to the genetic manipulation. This is a new method in the current context that some researchers have promised, and they have suggested that with the help of this new gene drive invasive method, they will be able to control the population of the disease causing mosquitoes. And at the same time, it is only done with the help of genetic manipulation and not by using any other extrusive methods, for example, insecticides, etc. Right? So first of all, what is the need of dealing with mosquito-borne disease? Mosquitoes are responsible for basically transmitting a lot of life-threatening diseases. Some of them are malaria, the Zika disease, lymphatic filariasis, the dengue fever. Most of them are basically coming from mosquitoes. From prelims point of view, please remember there are 2 species of mosquitoes which causes most amount of disease. First one is the female Anopheles mosquito. And the second one is Aedes aegypti. Aedes aegypti species. Question could come to you that which of the following disease is caused by which species of mosquito? To remember it better, take this trick away. So, in the alphabetical order, we write B, C, D, we write Y, and we write Z. So, chikungunya, dengue, yellow fever, and your Zika along with lymphatic filariasis. Please remember this. All of them are caused by Aedes aegypti mosquito. Only malaria is caused by Anopheles mosquito, female Anopheles mosquito, which basically carries a Plasmodium parasite. So, what is the villain over here? The Plasmodium parasite, which basically needs to be attacked. If we want to cure the disease, we are not restricting the population of mosquitoes. We are genetically modifying them in such a manner that the disease causing the problematic gene is cut out with the help of a special protein. These days we are using protein which is known as CRISPR-Cas9. But back in the days in 2003, a professor named Austin Bird developed this technology where he wanted to manipulate the genes using a special protein. By manipulating the genes, first of all, I am manipulating their reproduction. So basically controlling the reproduction of mosquitoes, and second, also manipulating the ability of the parasite inside them, the infectious parasite inside them to multiply. Let's say in this case it is the Plasmodium. Okay, so the Plasmodium bacteria would not be able again to multiply inside them with the help of this technique. So he targeted two aspects. First of all, genetic manipulation, and second, inheritance manipulation. These two areas were targeted. So please remember, the gene drive technology calls for gene manipulation, gene editing, and also manipulation of the drive or the inheritance pattern as to how these genes are getting inherited. Generally, it happens in accordance to the laws of Mendelian, but which we also call as the Mendel's laws. But now this inheritance pattern will also be changed. So, for example, I have curated the genes of this mosquito. Now I want this mosquito to be reflected everywhere, but there are only 25% chances that it will be inherited. It might render my technology inefficient. So what I need is a very strong inheritance pattern. So gene drive technology also transmits, or also it kinds of manipulate the inheritance pattern. In this particular case, you can see that the modified gene is transmitted to all the offspring. So the possibility of success is 100%. That is why this is the most efficient method till now to control the population of the mosquitoes. Okay. Now, how does it take place? We'll understand. So we will take the problematic gene. We will take the entire allele first of all. Okay. Now, in this particular one, we will first of all classify what is the problematic gene. Around this, I will use a protein scissor. What are we going to use? According to today's advancement, it is CRISPR-Cas9. This is the protein scissor that we use. CRISPR with the help of protein Cas9. So this will be attached, and the problematic gene would be, I'm explaining very simply, so it will be cut out. This problematic gene would be cut out, and hence now the inherited ones would not be having these problematic disease causing genes in the body. But what if the particular mosquito itself doesn't contain the gene but gets infected by a parasite like Plasmodium? So in this particular case, the gene drive technology, let's say this is a mosquito. Now it cannot produce a lot of offspring. Why? Because that particular problematic gene is already cut down. All the other offsprings will be having same genes. But the other problem would be, What if this particular mosquito gets infected by Plasmodium parasite? Well, that is also taken care of by the gene drive technology. How? I've already told you, it also restricts the reproduction of the parasite. Why? It triggers, this gene drive technology introduces such a protein into the body of mosquito that they themselves release antimicrobial substances. Antimicrobial substances, for example, melatonin and megalin. The names are not very important for you, but please understand, these antimicrobial substances, when they are released in good quantity, they themselves kill down the parasite. So here, I'm also killing the parasite if at all it infects. So the parasites will also be killed. Plasmodium parasites will also be killed. This is the gene drive technology. Talking about the gene drive technology, this is one of the preventive measures. I'm controlling the population, I'm controlling them at the genetic level. But there are other methods also. Some of them are the curative methods. For example, spraying insecticides. For example, using mosquito nets and mosquito rackets, etc. But they are not very effective. Another very effective method was the Wolbachia method. Wolbachia method was also used, and this was asked in UPSC Prelims as well. Now here, the Wolbachia bacteria is made to infect the female Aedes aegypti mosquito. Upon getting infected, it is now left into the wild, where it will be breeding with the other mosquitoes. Naturally, the offsprings will also be having this natural bacteria in them. What is the purpose of this bacteria? It reduces the reproduction rates. It also reduces the reproduction rates of the Plasmodium parasite that is infecting the mosquitoes. So both of them can be reduced. So in order to control the population, there are two methods, the gene drive technology and the Wolbachia method. You must remember, for understanding how to deal with malaria, especially in tropical countries like India, and how to deal with other mosquito-borne disease. Do not forget to code these two technologies in your GS3 papers and also in the paper where communicable disease is asked. Okay. The next point that we are going to cover, this is from the mains point of view. If you are asked about this technology, then what is the benefit of this technology? As you can see that I'm working on a genetic mode, which means it is a self-perpetuating method, which means I do not, unlike applying insecticides again and again, I do not need to have that. So this is more sustainable method. It reduces the need of frequent application. It is self-perpetuating in nature. So therefore, it is more cost effective. Of course, there will be one-time cost, but eventually it will be able to take care of the larger problem in longer run, right? Next, it also helps me to reduce the chemical pollution. The chemicals, for example, the mosquito repellent creams, for example, the pyrethroids. These are the chemicals which are used to shoo away the mosquitoes, kill the mosquitoes, the pyrethroids. And the other sprays, the other insecticides, they would not be used. Why? Because first of all, they not only leach into the water, also into the soil, but also enters into our food chain, and therefore causes a lot of biological damage as well. So therefore, it reduces the chemical pollution. It also reduces the biological pollution. Why? If you use something like a Wolbachia method, Wolbachia is highly intrusive. You are basically fading away the genetic diversity and purity by entering a bacteria into it. And this can also lead to damage of many non-targeted species. For example, some honeybees, bumblebees, etc. Or somebody who can consume these mosquitoes. On the other hand, this is a more targeted approach. I'm only targeting the problematic mosquito by genetically modifying them. So this also reduces the political pollution. Next, it does not also causes the health concern as compared to the other methods which are used as mosquito repellents. So of course, there will be lesser cardiovascular diseases, lesser soil pollution, water pollution, chemical pollution, biological pollution. So there are multiple benefits. Of course, there are certain ethical concerns with regards to genetic modification of any organism for that matter. But apart from that, there are multiple benefits which are written over here. Now, taking you to the PYQ. In the year 2023, it was asked, Wolbachia method is sometimes talked about with reference to which one of the following? The correct answer obviously is 1, controlling the viral diseases spread by the mosquito. But please tell me, what is Wolbachia? What does Wolbachia stands for? That you try to answer in the comment section. Alright, now let's move to the next topic. Now this particular topic is very interesting. What if you had only one lunch and you are very hungry? And yet you choose to give it to somebody who was more hungry than you at the cost of your own hunger. What if you are willing to share your notes among the other aspirants? What if you are willing to share your hard-earned money while you're already hard on your pockets, and you're able to share that money to the poor people? That behavioral characteristic is known as selfless behavior, but in the ethical terms we call it altruism. What are we calling it? We call it altruism. Nature and many other organisms also show some of the relationships. You must have heard about the symbiotic relationship. You must have heard about the parasitic relationship. But have you also heard about the altruistic form of relationship? Altruistic relationship is one in which a particular organism causes good to somebody else at the cost of themselves sometimes. This is the altruistic behavior, and researchers have recently found that some of the organisms, in fact, an amoeba of the name Dictyostelium, it shows a very natural altruism in the nature by helping the other organisms out. And this particular quality is inherited in a particular gene. This is known as the green beard genes. It's very funny, we are calling it green beard because upon microscopic analysis it was observed that that comes off as if these amoebas have grown a lot of beard-like tentacles, beard-like hairs. So therefore, if you see any such amoeba helping out another similar looking amoeba, this process is known as a natural altruistic relationship. Okay, so let's quickly learn about the green beard genes and the properties they show. So green beard genes show the following property. First of all, they have certain distinct trait. They have a distinctive trait. Not all amoebas, not all genes would be able to showcase these properties. Only certain having distinctive trait, for example, having green beard, only they will be able to show this trait. And the second one is like helps like, which means a green bearded amoeba or organism would be able to help another green bearded organism. So basically, I will only help you if you are similar looking to me. Even at the cost of me, even if I'll die helping you out, but I will do that. That is the second characteristic which they show. Third is, they also inherit altruism in their generations. Because in a room of many people, I'm only creating altruistic relationship with alike people having same distinctive trait. For example, green beards. I will be creating offsprings that will also have the same trait. For example, this one is always willing to give his lunch to somebody who's hungry. But he will only help that person who's also similarly willing to give off his lunch when somebody is hungry. He will not give it to some villain-like character. So they will create an offspring who will also develop the quality of sharing his lunch when the other person is hungry. That is the natural altruism showed by green beard genes. So for the examination point of view, it is not very important, but to have a basic awareness about these genes would help you out. And also it was a very interesting topic for me to teach. Now moving on to the next one. Our next topic is xylitol. Xylitol has been in news because recently the researchers of IIT Guwahati have created something which can act as a very low calorie sugar substitute. And this one is xylitol. It is not that they are the first one to discover it, but they discovered it from biomass, from waste. And that is why creating a more sustainable mode of production for xylitol. The second context which follows up is a depressing news. Eventually, a lot of usages of xylitol can also cause disruption in the production of platelet in the body, resulting into blood clotting inside the vessels, could also resulting into cardiovascular diseases. So, because of these two contexts, we'll be reading about this new compound, xylitol.
So you must have consumed some sugar-free chewing gums. You must have consumed some dental cares which are promising that there's no sugar, it's sugar-free. You must have consumed diet cokes. What actually is the component which is tasting sweet but rendered as sugar-free? These are the molecules, these are the compounds which are having low calorie content but provide sweetening effect. And these are known as the artificial sweeteners. These artificial sweeteners can be derived from both natural processes and artificial processes, but xylitol is one among them. This is one among the artificial sweetener, which is also known as diet, or which is also called as the sugar free. So for example, there is Xylitol impact gums, there are xylo sweets, a kind of artificial sweetener. So xylitol, as the name suggests, comprises of a suffix -ol, like alcohol. So basically, this is a sugar alcohol. First of all, it is a sugar alcohol. Alcohol, when it reaches the stomach, it can cause multiple side effects, for example, causing hyperacidity. That is one among them. And it also disrupts with the production of platelet in the body, which are responsible for clotting of the blood. So these are two harmful impacts of them on the human body, on the organisms. But other than that, when it comes to xylitol, xylitol is a sugar alcohol, and sugar alcohol has got multiple benefits as well. What are the benefits? Let's look into it. The first one, it has got a low calorie content. When compared with sugar, it has got a content 40% lesser as compared to sugar. So it is very good for people who are diet conscious, who want to lose the weight. So for them, this low calorie xylitol could be a good substitute. The second thing is, it is also good because, see, it is made up of alcohol, and alcohol is created out of a method of fermentation. So alcohol inherits a lot of bacteria. So therefore, they also supports the gut health. Gut health, of course, to some extent when consumed in appropriate proportions. So it also supports gut health by promoting the good bacteria and killing the bad bacteria. Okay. The third one is the production. Now it can be widely available because the production can be done both naturally and artificially, since it's an alcohol only. So the alcohols are generally created with the help of wood distillation. In this particular case, birch wood is used. And sometimes corns can also be used in order to create xylitol. But the best part is, it can also be created with the help of biomass. It can also be created with the help of biomass. How? With the help of intervention of certain enzymes and bacteria. What is the production process? I'll very quickly tell it to you. So let's say you have a biomass. Okay, you have like a pile of vegetable peels, fruit peels. Okay, and this needs to be decomposed. So now I can first of all convert it into glucose. This glucose will be named as xylose if I add certain catalyst to it. Okay. So this will be converted into glycose. Xylose is a form of sugar. Now, upon adding, this is catalyst or enzyme. Now, upon adding certain bacteria to it, I will be able to get xylitol. So what I'm doing fermentation. Fermentation is done with the help of bacteria, culture of bacteria, with the help of which I'm getting xylitol. So in both the ways, it can be created. So the production is very easy, and it is more affordable as compared to other forms of artificial sweeteners. Then the next one is, it can be used for the people who are suffering from diabetes or from high spike of sugars, because it has got lower concentration of sugar, and it also dissolves and gets digested into monosaccharides very easily. So therefore, it can also be used for the diabetic patients. It can also be used to fight against the ear infection and also against the tooth decay, because it actually promotes the development of calcium, and that is why it also supports the bone density. It enhances the bone density. So it prevents tooth decay, bone density increases. It also helps
in the hygiene of ear by removing the detrimental bacteria. And next, since it also releases sugar in a very controlled amount, so it doesn't cause a sharp spike in the sugar levels in the body, which can cause many issues, for example anxiety, indigestion, for example diabetic situations, etc. So therefore it is also good in controlling the sugar spikes. So it is more or less, it is advisable for you to consume products which are containing xylitol, but of course in regulated amounts, because now you also know what are the disadvantages of the same. Moving on to the next topic. The next topic is about the craters on the Mars surface that have been interestingly discovered by Indians and named after Indians. So recently, the Physical Research Laboratory, PSL, where is it headquartered? It is headquartered in Ahmedabad. It was founded on the year of independence, that is 1947. Founded by Dr. Vikram Sarabhai. And is it a part of ISRO? It is a part of Intergovernmental Organization of Department of Space. So this Physical Research Laboratory has recently discovered 3 craters on the surface of planet Mars. How was it able to do this? With the help of radar, indigenized radar, that is known as the Sharad radar. The full form is not very important to you, but please remember it was made with the help of, this discovery was made with the help of Sharad radar. Was it mounted on Mangalyaan mission? No. It was mounted on the Mars Reconnaissance Orbiter mission of NASA, which is still exploring the surface of Mars, and this particular radar was mounted on it. Okay, and with the help of this radar, they got to discover 3 craters. Can you see this big crater of about 65 kilometers diameter? This is the Lal crater, and 2 other small craters, the Mursan crater and the Hilsa crater. This Lal crater, Mursan and Hilsa, was India alone to give it the name? No. In order to give name to any astronomical object, you have to get the permission of the International Astronomical Union, which was founded around the time of World War I, and it is currently headquartered in Paris, France, with the objective of enabling international cooperation in the astronomy. So it is only the International Astronomical Association Union which will be able to provide the name, to legalize the name of any astronomical surface. But by the way, the names are Lal because of the Devendra Lal, who was the former director of Physical Research Laboratory and was also a very prominent geoscientist. The second one is Mursan, which is towards the east. This is a town in Uttar Pradesh. And Hilsa, this is a town in Bihar. So they have been given indigenized names. Now from the prelims point of view, we need to know about certain Mars mission and what are the space agencies behind them. So let's have a look. Here's a list of some of the space missions on Mars, and to know about the space agencies that have been operating it, it's very important. So first of all, ExoMars, this is by European Space Agency. See, why am I telling you this? Because such questions have been asked very frequently in the state PCS examination. So to know about these facts, because of the growing Mars frenzy, becomes very important. Now next is Tianwen. As the name suggests, it is Chinese mission. So it is by Chinese Space Agency. Hope mission, very interestingly, is by United Arab Emirates. So this is by UAE. Okay. Next is Curiosity, Insights, Perseverance mission. Most of the missions are basically from NASA only, but there is distinction between them. Insights mission is a lander mission. Okay, Insights is a lander mission. Curiosity and Perseverance missions are the rover missions on basically the surface of Mars, considered to have achieved highest sophistication in terms of exploring Mars. Okay, Curiosity, Perseverance, Insights, all of them by NASA. Again, some of the orbiters are there. Recently, in this particular one, we have read about MRO, which is Mars Reconnaissance Orbiter. So MRO and MAVEN, both of them are the orbiters around Mars, again by NASA. And what about Russia? It's a no-brainer. They have created certain satellites in the name of Mars 2 and Mars 3 only. So nothing much to learn on that, but please make sure that you are creating notes of these missions and their space agencies. With this topic, we will understand how again India and NASA have collaborated to have research and studies upon a very interesting bacteria, and this bacteria is Enterobacter bugandensis. Why is it a very interesting bacteria? Because it is so resilient and resistant. It could thrive in the space as well. So a lot of the strains were found in the International Space Station, which is located in the low Earth orbit. Talking about the International Space Station from the prelims point of view, do you know which are the countries which manages the ISS right now? Let me tell you that first. So, two are coming from the western areas, Canada, USA. One is European Union. Coming from the central region, and coming from the eastern region are Russia and Japan. So currently these 5 space agencies are managing the International Space Station. I hope this is clear. Now let's have our focus towards this particular bacteria. What does the name suggest? That this is Enterobacter. It's a bacteria. Okay, showing morphological features. First of all, that it is rod-like in shape. So it has got rod shape, as you can see. This is a rod-shaped bacteria. I've already told you in the previous month's recital that bacteria can be classified in accordance with their shapes. It could be a coccus bacteria, spherical in shape. It could be a flagella, a spiral-like bacteria. It could be a rod-shaped like bacteria. This particular one is a rod-shaped bacteria which shows characteristics of being gram-negative. I have also talked about what is gram-negative. I will again make you revise on this. So first of all, some of the bacteria can be divided into 2 classifications based on their cell walls. So if the cell wall is very thick, then in that particular case they are known as the gram-positive bacteria. Why? Because if I stain this particular bacterial strain with a particular reagent, basically with a gram stain, it will still retain the color of purple. So if it retains the color of purple, then it is known as a gram-positive bacteria. Why? Because it has got a very thick wall. Okay. The second one is having a thin wall. And what it does is, it is not able to retain the purple color entirely, rather it stains into a faint pink color. Why? Because it has got a thin cell wall, and this is known as a gram-negative bacteria. You may ask that my concern is, ma'am, which of them are more detrimental? The more infectious, the more dangerous kind of bacteria, the more harmful one are the gram-negative bacteria. Because of thin cell wall, they can have contagion, or they can infect a particular agent very easily. So therefore, the more problematic one would be, harmful one would be the gram-negative, and this one is a gram-negative bacteria. The third property is it shows high degree of resilience. Shows high degree of resilience because it is available across all the ecosystems. It could, where is it basically found? In the feces, in the soil, in the water bodies, in places where waste treatments are not done properly. So generally, these bacteria are coming from such areas, which, by the way, can also enter to us in the food chain with the help of oral fecal route. Now, if India, and from the side of India, IIT Madras has collaborated with NASA and both of them have researched upon this particular resilient bacteria. The reason for this is, if I am able to research on this, I will also be able to gain insights as to how certain kind of microbes or microorganisms are able to generate lot of resistance, which will eventually help me to understand how AMR, antimicrobial resistance, is taking place. So in order to deeply study on this, so when I am researching on this Enterobacter, it will help me not only to understand the AMR better, the AMR situation better, but it will also help me to understand what are the prospects of these microorganisms which can thrive in space and can suggest life in space. Okay, this was all about this particular topic. Also, please remember, this is a rod-shaped bacteria. Had the name been Enterococcus, then it would be spherical in shape. Okay, but wherever you see the term bacillus, for example, lactobacillus, all of them are rod-like shaped. Okay. Now, since antimicrobial resistance is such a hot topic for the UPSC exam, I want you to prepare way ahead for it. So for that, I have two homeworks for you. First one is regarding a mains question, where you have to discuss the global implications of multi-drug resistant pathogen and highlight the role of international cooperation in addressing this critical health issue. And second one is from a PYQ asked in 2019. Four statements are there, and you have to answer which of the following are correct with regards to how a person develops a multi-drug resistance. So have a look into this particular slide and try to answer it. In the month of June, Japan saw an health emergency. Why? Because more than 1,000 cases were reported of people having this very toxic disease, very deadly disease, the streptococcal toxic shock syndrome. Okay, first of all, let's get the basics correct. Tells to me that this is a streptococcal disease. Streptococcal is a virus, a fungi, a parasite, or a bacteria, you tell me. Streptococcal is a bacteria. So this is first of all a bacterial disease. And you must have already heard about streptococcal a lot of time, because anytime anybody gets a strep throat, that is because of streptococcal, but it's not very deadly as such. But there is one particular species of streptococcal bacteria which is known as gas. The group A streptococcal bacteria. Now, this has the ability to multiply in a large amount of numbers and eventually produce enormous amount of toxins in the body when contaminated for a longer period of time. And that is why it not only affects just the respiratory tract, but also sometimes impact the urinary tract, sometimes also penetrate deep inside the bloodstream of the body. And as a result, they can cause some mild symptoms, for example, fatigue, for example, fever, for example, low blood pressure, etc. And eventually can lead to organ damage as well. Why? Because of massive release of toxin substances. And that is why this is known as a toxic shock, because they can rapidly multiply and produce a lot of toxin in the body in a large amount of number. Okay, this is a toxic shock syndrome. Of course, talking about the morphology, this is a coccal bacteria, which means it is spherical in shape. And I have already told you that those which are spherical in shape, those who are coccal, they are the gram-positive bacteria. These are the gram-positive bacteria. The last one which we have read, Enterobacillus, was a gram-negative bacteria. Okay. Now, talking about this syndrome, how does this bacteria originate, or from which places does it come from? So it can originate from the direct contact of the infected person. So it is the contagion rate is very, very high, highly contagious bacteria. This is a syndrome, and this particular one we have read in regards to the communicable disease in the part of GS3 of the syllabus. And it affected which country? Important for state pieces. This is infected Japan. Okay. Now, let me also bring some of the NCERT references, since we've read about a lot of bacteria, their shapes and their classification in this particular month of recital. The NCERT specifically talk about the bacteriophage. That is why UPSC has asked on this. They have talked about classification on the basis of shapes. So this is the bacillus bacteria, this is the coccus bacteria, this is the flagella bacteria. Alright, Then have you also heard about adenoviruses? Question which was asked in UPSC, I've already showed you in the first topic of the first slide. So these adenoviruses, which are basically DNA viruses, they are also there in NCERT. So therefore reading NCERT becomes very important, especially at least till 10th standard. Okay. Then with regards to Streptococcus, we've already read that it causes strep throat, it causes a toxic shock syndrome, but it also has certain significances. For example, with respect to NCERT, they have said that a kind of streptokinase, a protein, an enzyme. So it's an enzyme. AS is written, right? Enzyme which is produced by the Streptococcus bacteria. And when it is modified by the genetic engineering, it can be used as a clot buster for removing the clots from the blood vessels of patient. So there are positive impacts of the bacteria as well, of the same bacteria, Streptococcus. Okay, depending upon how is it genetically modified and to which amount it has infected a particular part of body. So this is the NCERT reference, which basically suggests to me what is the importance of reading NCERTs, because a lot of conceptual question that comes in prelims examination and also in the mains examination can be directly answered and predicted with the help of NCERT reading. So keep your preparation simple, but at the same time, be very disciplined about your approach towards NCERT and some of the legitimate book sources. Okay. Do you know what is the quality of a bad alcohol? It has lower alcoholic content. But some of the producers of the cheap alcohol, they add a substance into this alcohol, the consumable alcohol, in order to increase this alcoholic content, that too in a more affordable price. They add a substance which is known as methanol. Methanol, which is highly inflammable, which is also something which creates a lot of acid, is basically used in order to create cheaper alcohol. So therefore these alcohols are also known as the fake liquors, the tainted liquors, or often called as the bootleg liquors. Bootleg liquor has been in news. Bootleg alcohol has been in news in the month of June because it took the lives of more than 100 people in the southern states of Kerala and Tamil Nadu. Why? Because they consumed it and eventually they died. So let's first understand what is the substance of methanol and why methanol is one of the hot topics in UPSC, especially when it comes to generating sustainable fuel. First of all, let me draw the diagram of methanol. It is the simplest form of alcohol. When I am saying this, it contains only one carbon atom, 3 bonds of hydrogen and 1 bond of hydroxyl group, which basically provides it the alcoholic content. We have already learned about xylitol. Xylitol has application in the form of a replacement of sweet, a replacement of sugar, and this one has got application of generating a very efficient fuel. Okay, so methanol is also used in fuel. Now, looking into the structure, can I say that it has got low carbon content? It has got low carbon content because only one molecule of carbon is there. So can I also say that upon burning it as a fuel, I will be able to generate lesser amount of greenhouse gases, for example, methane and carbon dioxide? Yes. So one of the application is that it is a more greener fuel as compared to our diesels and the other gasolines. Why? Because lower amount of greenhouse gases. Then, can I also see that there are 3 bonds of hydrogen? And I know that hydrogen is very good for bringing energy efficiency, and that is why hydrogen fuel is there in use, right? So we are trying to pull in hydrogen fuels and we are also trying to pull in the hydrogen economy. So for that, these 3 bonds of hydrogen will help us, right? So when I have hydrogen, it increases the energy efficiency of this fuel. So energy efficiency is also high. Next feature about methanol. It is very easily produced. It can be produced from the fossil fuels, for example, primarily, and it is best produces from coal and natural gases. Even better and even greener production would be done with the help of natural gas. But it can also be surprisingly created with the help of biomass, with the help of municipal solid waste. So, the production is also easy. When I am saying production is easy, it will also help me to facilitate waste management, because waste can now be fermented and then it can be converted into methanol, which can be used in the fuel form, right? Talking about its talking about this natural production process, as we have read that it is generally, the alcohols are generally a product of the distillation of wood. So similarly, this is also produced from wood distillation. Therefore, this is also known as the wood alcohol. It is also known as wood alcohol because produced with the help of wood distillation. Xylitol was also produced with the help of which wood? Birch wood. Okay, distillation of fermentation of birch wood. Okay, so basically it can help me to manage the biomass, and when I'm able to create it with the help of waste and biomass, it also comes to me as if this is a more affordable form of fuel. It is a more affordable form of fuel, and in order to create it, do I need to import it from anywhere? Not at all, because I can create it with the help of biomass, with the help of waste, with the help of fuels, with the help of existing coal reserves I have, natural gas reserves I have. So naturally, my import dependence on the fuel will also be reduced, which is basically an agenda of Indian economy, right? So it will also help me to reduce the import bill. Reduce import bill. You ask, ma'am, how much? Well, it is estimated by Niti Aayog that if we blend with the gasoline 15% of methanol, then that 15% of blend would reduce the cost up to 15% on the consumer. So it is a 15:15 cost to benefit ratio. 15% blending, 15% reduction in the prices of the import bill. Fine. Now, I have already told that it is more eco-friendly, it is more sustainable form of fuel. When it is sustainable, it is also non-polluting in nature. It is also non-polluting in nature because can you see the formula over here? It is not very complex as compared to other sources of fuels, for example, coal, etc. So therefore, it will not be able to create particulate matter, it will not be able to create SOx. I don't find any sulfur here. I don't find any nitrate also here. So nitrous oxide will not be created, sulfur dioxide will not be created, particulate matters will not be created. Basically, the overall pollution or the overall greenhouse gas creation will be reduced up to 20% by using methanol. It is because of these reasons that Indian government is now putting impetus into production of methanol and building a methanol economy in India, especially for the fuel production. Not that it is not having another application. It is of course used in creating acetone and acetic acid and formaldehyde, some other industrial solvents. It's very important for industrial purposes as well. But most importantly, the need of the hour is to create biofuels, and methanol could be a pioneer in that. Okay. Talking about some of the physical properties of methanol, you must know that first of all, it is a colorless liquid and it is completely miscible in water. So therefore, it is used as a solvent in order to preserve things. So it is also very good preservative. It is preservative. It is also soluble, so it can be used as solvents in industries. Along with this, you should also know that it is highly flammable. The flash point at which this particular fuel will get burned or will catch the fire is 9 to 12 degrees centigrade. So it has to be maintained at a relatively higher temperature, and that is why we cannot promote the 100% use, though it is very much possible, but it cannot be used directly into the fuel engines. Why? Because the flash point is very, very low. Which also makes me take you to the next point. It has got anti-freeze properties. So in the places where there is very cold temperatures and the fuels of the driving cars, etc., or the windshield can get frozen up very quickly, in those places such methanol can be added in such solvents, in such fuels. Methanol can be added because the anti-freeze point is very, very low, which means it does not get frozen up very quickly. It gets frozen up at minus 80 degrees centigrade. Alright, now from the mains point of view, we will quickly analyze the advantages and the disadvantages. I've already talked about the advantages with the help of the structure, molecular structure. But to summarize it again, it helps us to reduce the import bill by 20%. It helps us to reduce the greenhouse gas emission. It helps us in fuel diversification. It can not only be used in the marine fuels, in the automobile fuels, in the aviation fuels, but also in the LPG gases. In fact, The Assam government has already promoted Assam Pharmaceuticals, and the Assam one particular company of Assam has promoted the use of or the replacement of LPG gases with the help with this methanol. So it can help us to abate the air pollution. It can help us to diversify our fuels better. It also helps in generating a lot of job because it has got diversified application. A lot of industries would be benefiting from it since it's an emerging technology, upcoming biofuel. So therefore it will be able to create as much as 50 crore jobs in India, right? Then, what are the disadvantages? First and foremost, what is the production? Production can be made through different processes, different sources. But what is one viable source of production so that I'll be able to bring economies of scale? Good amount of methanol can be created. That can be done with the help of natural gas. Do you know how much amount of natural gas does India have? Well, we are already having certain crunch when it comes to natural gas reserves. We are importing it from other countries, and now if I want to divert it towards generation of methanol, that would also be a problem. So lack of domestic natural gas is one thing. Second is we could also create it from the coal reserves, but this will not help me create the greener form of methanol because the coal reserves are not of that good quality in India, so they will be rather polluting. The second is lack of infrastructure. How to bring or how to build such distilleries or such industries which can promote conversion of methanol. For that, we do not have adequate policy support in the terms of finances that we are getting in methanol industry. Lack of incentivization, lack of infrastructure is there. Okay. Next is lack of acceptance. I've already told you this one is highly inflammable. So even if government wants to have a higher percentage of blend, so for example, Niti Aayog's methanol program suggests that there should be 20% blending of methanol into the existing gasoline, and the variant of methanol would be dimethyl ether. So it will be added in 20% blend, but people are still reluctant towards it because they feel that it might be unsafe to use methanol. They are fine by consuming methanol, ingesting them, but when it comes to using them in the fuels, in the cars, they are reluctant towards it. So there's lack of awareness, there's lack of acceptance among the people. If we are able to cater and deal with all of these challenges in the near time with the help of our policy intent and associated content, proper content, we will be able to shift gradually towards the methanol economy. The next topic is Claude Sonnet. Claude Sonnet, have you ever heard of it? So anyway, have you heard about ChatGPT? Have you heard about Bard AI? What are these? These are generative AI models which are based upon large language models, okay, which use neural networks, which uses artificial neural network, which uses deep learning, generative AI intelligence. All of this we have discussed umpteen number of times, and because of paucity of time, I wouldn't be able to explain it a lot over here, but Claude is a very similar variant of a large language model which is developed by a company which is named as Anthropic. So why is this in news? Because when its report card was compared with the existing technologies, the existing softwares like the ChatGPT and the Bard AI of Google, giants like these, it was found to be performing much better. You can see that it has outperformed ChatGPT. In fact, the most latest version of ChatGPT, most latest version of Gemini, it has outperformed all of them. What is the report card? It's very interesting. So let's have a look. Graduate level reasoning, it has got 60% of graduate level reasoning. Undergraduate level knowledge, 88%. Code human level, 92%. It can have multilingualism up to 91%. It can have mixed evaluations up to 93%. Can crack the school maths of 8th grade up to 96%. So basically it is very closer to the general artificial intelligence and not just generative artificial intelligence. This is the Claude Sonnet of Anthropic. Now our last topic for the day is about superhydrophobic catalyst. Very recently in the month of June, an article was published by a team of scientists, including some of the scientists from our Assam. So they have discovered a new kind of catalyst in order to make the production of biodiesels more affordable. And this is with the help of a newly created superhydrophobic active carbon catalyst. Now from UPSC point of view, except for understanding how to make the biodiesel more affordable, the rest part of it is not very important, but I'll just let you know about the interesting stuff. So have you seen a lotus leaf? Lotus leaf has a natural property of creating droplets of water around it. Why? Because it is showing water repellent property. This water repellent characteristic is known as hydrophobic. Phobic means fear. Phobic means fear, and the fear is here of water. So substances like these can be used in a lot of industrial applications. So basically these substances do not react with water. They are not bothered by the water because they repel water like anything. In this particular case, the substance that we are using is activated carbon, a form of carbon which is created, which is basically converted into very small pores, so that the surface area exponentially increases, and therefore the contact angle with the water decreases, so water cannot really touch it, and therefore whenever water tries to come, it basically gets reduced into a little into a shape of very spherical water droplet, but it cannot wet this particular substance. Now, talking about the biodiesel, what is the difference between an oil and what is the difference between a biodiesel or a diesel? So please understand the mechanics behind it. So oil is nothing, vegetable oil is nothing but a long chain of carbon. A long chain of carbon. If somehow you are able to break this carbon chain into 1/3, if you are able to break this carbon chain into 1/3, then you will be able to get a very important fuel, that is a biodiesel. How to break it? There are 3 processes. One is to apply heat, to apply high temperature, which is known as pyrolysis. So the first process of breaking is pyrolysis. Second is application of large amount of pressure. This is known as the process of cracking. Second is cracking. Third is by adding two bonds of hydrogen. By adding 2 bonds of hydrogen, and this is known as the process of hydrogenation, the most utilized method to be used in the industrial processes. One of the shortcomings of hydrogenation is that it produces a lot of water. When this particular product is affected by a lot of water, and especially what happens is when you have to break this long chain, you have to apply a lot of catalyst which can speedify the entire reaction. But the conventional reactions, the conventional catalysts that we have been using are acids, strong acids. For example, the conventional catalysts that we have been using are strong bases. For example, sodium and potassium hydroxide. So we are using this. When they react with such a chain, they produces a lot of water. And many of the reactants then, or the speed of the reaction eventually lowers down because of this water. This makes the entire process very costly because the amount of biodiesel that I am creating from, let's say, 1 kilograms of vegetable oil would be lesser because of water. So what if I create a catalyst which is repellent to water? That will expedite the process, that will create more amount of product, and that is basically the superhydrophobicity, and this is the superhydrophobic catalyst. This is basically used in biodiesel production. Okay, so this was all about Recitals discussion on science and tech. I hope you've enjoyed it. Science is a very interesting subject given your basics are good. In order to get your basics right, please read the NCRTs, please read the foundational notes that you are being given, and also make sure that you are thorough with the current affairs. If in case you come across any other difficulty, feel free to reach out. Thank you so much and have a nice day.